So my name is Martina. I'm a first-year PhD student within Cox-Maros group in the Quadram Institute and Earlham Institute in Norwich. And in collaboration with Lindsay Hall, I studied the effect of bifidobacteria on intestinal bowel diseases such as Crohn's disease. Thanks to the expertise that I have in my lab in organoid technology, but also multi-omics approaches, I'm in a very unique position because I can predict interactions between bifidobacteria and intestinal cells and then validate them directly in the lab using organoids technology to validate my initial hypothesis. We know that bifidobacteria can interact with intestinal cells and modulate their cell function. However, we don't know what are the specific modulating factors. In our lab, we aim to study how bifidobacteria interact with intestinal cells, and this is key to understand the beneficial effect of bifidobacteria for gut health. We are interested particularly in studying one subtype of intestinal cells called Paneth cells, which are really important because they release antimicrobial peptides in the gut to fight against infection. And their function is generally changed during inflammatory bowel disease. Gut organoids represent a very important model to study Paneth cells because Paneth cells cannot be cultured in isolation in the lab. And at the moment we are using 2D organoids to study how bifidobacteria interact with intestinal cells, in particular Paneth cells, in the apical side. In our group, we use intestinal organoids to generate cell-specific transcriptomics and proteomics data, which we use to understand which molecular pathways are regulated by bifidobacteria, for example, in Paneth cells. And these are very useful because then we can see which pathways are modulated and we can test them in the lab to validate our initial hypothesis. And by using this method, we hope to find out which pathways are modulated by bifidobacteria, in particular which ones are dysregulated in inflammatory bowel disease. And this will help us understand how we can use bifidobacteria to restore the dysregulated pathways. Thanks to our close collaboration with the NHS endoscopy unit within the Quadram Institute, in the future we will be able to make patient-derived human gut organoids. And this is very exciting because we can not only study the effect of microbial factors, but also the effect of host genetics on the pathogenesis of inflammatory bowel disease in a patient-specific manner. We believe that the successful development of human-derived 2D gut organoids will help us understand how bifidobacteria modulate the function in intestinal cells during intestinal bowel diseases and hopefully pave the way for new translational developments and cure for these pathologies.